Today's lecture is on microbial growth. So microbial growth is cell division that produces new daughter cells and increases the total cell population. So cells living in a biofilm will communicate and collaborate in order to survive. So in a healthcare setting, these are important because they're difficult to treat and they do contribute to persistent types of infections. So what is a biofilm? It's a community of mixed microbes living together. So one of the things we look at when we look at microbial growth is what's called generation time. And that's the time it takes for a cell to divide. So the average range then would be anywhere from 15 minutes to about 24 hours. And it depends on the species and conditions. So bacteria like E. coli and staph, for instance, usually replicate every 20 to 30 minutes. So bacteria have 4 distinct growth phases, and we're going to go through those. And when we culture them, we have a lag phase, and that's when they adjust to their environment. Exponential growth phase, that's when they're actively growing. And then stationary phase is when the number of new cells equals the number of dying cells, and then eventually they're going to die. So please know these 4 phases. Phase 1 is the lag phase, and that's, again, when the cells adjust to their environment. Then we have phase 2, which is considered log phase or exponential growth phase. So we can use those two terms interchangeably, and that's when they're actively dividing. The stationary phase, that's when the growth rate levels off, the new cells equal the cells that are dying off. And then in the death phase, as you can imagine, that's when the cells are starting to die because there's no nutrients left, there might be toxins building up, etc. So what factors affect growth? Well, temperature affects growth, and one of the things we want to keep in mind is that lower temperatures below their range are going to slow things down. But very high temperatures will denature cell proteins, and that ultimately kills the cells. So we can have a maximum, and that's the highest temperature that will support growth. Minimum, lowest temperature that supports growth. And optimal, that's the temperature where the cellular growth is the highest. So these are some terms you want to know for your exam. First one, psychrophile. Please know they grow between -20 and 10 degrees Celsius. Psychrotrophs will grow about 0 to 30 degrees C, and these are the ones that are associated with foodborne illnesses. They can actually grow in your refrigerator. And mesophiles, they grow anywhere from 10 to 50 degrees C, and these are associated most with pathogens. They grow very well at 37 degrees C, which is our body temperature. Thermophiles, thermal, think hot. They grow anywhere from 40 to 75 degrees C. And lastly, guys, extreme thermophiles, these are not ones we would worry about as far as infections go. They grow 65 to 120 degrees Celsius. pH is also a factor that can affect growth. So we have 3 groups that we can discuss. Acidophiles, they grow in a pH below 5. So they like an acidic environment. Neutral or neutrophiles, they will grow best between 5 and 8. And if you think about it, our pH inside of our body is about 7.2, 7.4, somewhere around there. So these are the ones that make up the majority of the microorganisms that we are concerned with when it comes to an infection. Alkalophiles like alkaline environments, so they grow best 9 to 11. Salt conditions are something we need to consider as well, and halophiles will thrive in high salt concentrations. Some of them tolerate all the way up to 35% NaCl, so pretty salty environments. Now, oxygen is something that is also a requirement for growth, so we'll walk through some categories for microbes based on how well they grow in oxygen or whether they grow in oxygen at all. So microbes that depend on oxygen for growth need to detoxify it. So please know that these two enzymes, superoxide dismutase and catalase, will detoxify harmful oxygen species. They'll actually detoxify it. So again, superoxide dismutase, catalase, detoxify harmful oxygen. So here's our categories. Please know obligate aerobes need oxygen to grow. So those would be ones that would be associated with skin types of infections, or if it's on the top of your body, it's exposed to a lot of oxygen. Microaerophiles will only use small amounts of oxygen. Now, facultative anaerobes will grow with and without oxygen, and they do use oxygen if it's present. And actually, they prefer the oxygen because they get more ATP, but they can grow without it as well. Aerotolerant anaerobes will tolerate oxygen, but they don't use it. They can deactivate the harmful oxygen, but they're not going to use the oxygen anyway. Now, obligate anaerobes do not use oxygen, and in fact, oxygen is toxic to them. And this just shows if you culture these in the lab, your obligate aerobes would grow at the top. The anaerobes tend to grow from the bottom up. Then microaerophiles find a spot where they have a little bit of oxygen. And our aerotolerant, they'll grow throughout the tube because they're not using the oxygen. The facultative anaerobes tend to cluster at the top, but when there's no oxygen for them to grow in, they'll grow anywhere in that tube. So there's various types of media, and in the lab we cover more of that, but the media can be liquid, solid, semi-solid, etc. So agar is what we use in our Petri plates, and sometimes we just put nutrients in there, sometimes indicators and that kind of thing. But they're used quite widely to culture different types of microbes. So if we have what's called a slant guise, as you can see, this greenish agar here is on a slant. And deeps are when you see like straight across here, but you would stab that to look for growth. And this is an example of a motility test to see if the microbe actually can swim. Now we can have differential media, and that's media formulated to visually distinguish one microbe from the other. So you might have a bacteria that will grow white on a media, but on that same media, you might see another one that's going to grow a different color. And that's based on what's in that media to differentiate between those two microbes. Now here's another example of differential media, and this is something in the lab that we use quite often. This is blood agar. Now, if bacteria can break down the red blood cells, it clears out the media. If it partially breaks it down, you kind of get a greenish precipitate and a little bit of lightening. And if it doesn't really do anything except for grow, it just will kind of blend in on that blood agar plate. Now, selective media is a little bit different. You will single out a bacteria that has a specific property, and what's in that media will promote the growth of certain bacteria, while at the same time it suppresses the growth of others. So whatever is in the media, again, will allow certain ones to grow, but it suppresses others so that they don't grow. And mannitol salt agar is an example of that. Now, in this particular case, the salt would select for any that cannot tolerate it. Now, both of these bacteria grow just fine because they tolerate the salt. However, the one on the left is fermenting. There's mannitol in there, which is a carbohydrate that gets fermented, and it turns the yellow color. So you can see that we're selecting for mannitol fermentation as well as salt tolerance. And ENB plates, you can actually select for gram-negative bacteria. They can be a strong fermenter, which would have this greenish color. moderate to low fermenters, the purple color. And if they don't ferment at all, they just kind of grow on the plate and blend in. Now, aseptic techniques are things that we discuss a lot in the lab, and as healthcare workers, you have to make sure that you're working in an environment that is sterile as possible. Plus, when you're collecting specimens, you want to make sure that they're collected in a sterile method. So they need to be collected and transported as aseptically as possible. You have to use sterile tubes, sterile containers. You should be washing your hands before and after specimen collection, etc. Wearing gloves is also very important as well. And once you collect, let's say, a urine specimen, you want to seal that right away so nothing else actually gets into it. Sometimes you'll have to refrigerate things also or ship on dry ice depending on what you're collecting. Now, one way then that the lab would take your specimens to isolate what's causing an infection is to do a streak plate. So please know, the purpose of the streak plate is to isolate colonies. And a colony is a group of cells arising from a single cell. Please take a screenshot of this and upload it to your Dropbox. So with the streak plate, you start to wipe very heavily in one direction. Then you go in another direction. Sometimes you go 3, sometimes 4 times. In the lab, we do a quadrant streak, so we actually go 4 times. But the idea is once you get down to the end, each individual dot here is a colony. And that arose from one cell. So it's a bunch of cells that arose from one cell. Now we have a lot of different ways to identify microbes. That's more something you need to know for the lab, but you're going to look at the shape, morphology of things like colonies, do some biochemical analysis. We don't in our micro labs. You're not required to do genetic testing, but that is done out there in the real world for sure. So how do you control then contamination? So you can decontaminate, and that will reduce microbial populations. Sterilize, that eliminates everything, all bacteria, viruses, endospores. And disinfect, and disinfection just reduces the number of microbes that are going to be present. Now, pasteurization is something we use for milk, and it doesn't sterilize the milk. What it does is you are administering high heat for a short period of time, and it will eliminate some of the pathogens that are in there. But that doesn't mean it gets rid of every possible bacteria. So please note pasteurization, high heat for a short time. What dry heat will do is it will actually sterilize or disinfect, and basically what you're doing is oxidizing. So eventually, if you apply this high heat for a long time, in the lab we actually inoculate our loops by putting them into a Bunsen burner. You'll sterilize it that way. Now we can also have chemicals, and some are considered germicides. Cide means to kill, so a microbiocidal will kill microbes. Some of the compounds will only inhibit growth. Those are considered microbiostatic. So static means it inhibits, it's not really killing the growth. So 2 key classes are disinfectants. Those are used to treat inanimate objects, and antiseptics. That's what you guys actually put on your skin, so on living tissue. So a little bit about some common ones that can be used. Alcohols, please know, will denature proteins and attack lipid membranes. So remember, all bacteria are going to have a plasma membrane, and gram-negative actually have an outer membrane as well. Phenols, those will destroy bacterial cell walls and interact with proteins. Some products that contain phenols would be some of the Lysol products out there actually have phenol in them. And if any of you have ever used Chloraseptic, that funny taste in there is actually phenol. Halogens, examples would be like iodine, chlorine. those will oxidize your cell components. Now, a very interesting type of sterilant out there is ethylene oxide. This is a colorless gas, so the benefit of this is it can get in all those nooks and crannies that scrubbing is not going to allow. So this will damage both proteins and nucleic acids.